We will start by showing how the dataset and the sequence file can look like. This is one of the files we will use as input in this tutorial. There are no limits to the number of columns or rows. There should be a column with the features, in this case the name column, and sample columns. These are at the end of the dataset, as we can see here. Column retention time should be included if we want to do merging and drift correction. The column labeled mass or mass to charge should be included for merging as well. Other requirements can be seen in documentation. Now let's take a look at the sequence file. So this file should include 7 columns, as you can see here. For the columns where no information is available, this can be left blank, like the paired column in this example. The samples in the first column should have exactly the same names as the ones in the data file. The order of the samples is used only for drift correction. The groups can only include letters and numbers, no spaces or special characters. If the samples are run in different batches, this can also be included in the batch column. Let's start by uploading our dataset. After uploading, the app goes through all the columns and labels them. You should make sure that labels are correct before proceeding. Labels like name, retention time, mass, and adduct should not be present more than one time. And we can see that everything looks fine, so we can now upload the sequence file. Again, make sure the sample names in this file match the sample names in the data file. Now let's check the information we uploaded and if it is assigned to the correct samples, and we can see that all the samples have the information that was in the sequence file. So now that everything is uploaded, we usually go to the Explore Data panel and we navigate through the different tabs. We can, for example, check for abnormal medians in the QC samples. We can look for outliers in this. In the feature viewer, we can look for internal standards, which we have in this dataset. These are identified by the app by including IS in parentheses in the name of the features, and we can see them here. Now we will start with our workflow. The first step is blank filtration. When doing this, we normally keep the app in the Summary tab because we can see the changes to the CV straight away. 5 times above blank is usually the signal that we use. Typically, this removes around 10% of the features, but this also depends on the extraction of the samples. If it seems like it removes too much, just try it with a smaller value. After this, we usually run a quick PCA to check the data. Here you can see there are 2 panels and you can compare before and after. Next, we do the missing value filtration, and here we have 3 different options. We can do it to the entire data set, we can do it just for the QCs, or in group. And we will start by removing the features where there are less than 80% measured values in the QC samples. And we will also use the in-group filtration, but with a lower threshold, usually around 50. This setting goes through all the groups and makes sure that the feature is above the threshold in at least one of them. Again, we run a PCA to check for changes. We can see that the CV decreased. Remember to make sure you click Run PCA each time because it does not update automatically even if you select different datasets. Now, since we have internal standards in this dataset, we will do internal standard normalization. Generally, we run it one by one to see how each of them affects our data. If there are missing values in the internal standards, this will not work. However, when selecting more internal standards, there should be no problem. This process takes a bit of time, so we'll fast forward through it. One thing to bear in mind during this is that the CV in the QCs should not increase. So we are going through the first one. We will run PCA after each one, but without saving the data. We can see that the CV decreased, so this is one that we want to use in the final normalization. For the second internal standard, we can also see a decrease in the CV of the QC samples. We will keep going through it. Third one also, we can see a decrease. For the fourth, we also see a decrease. Now on to PE. PE doesn't change much, so we probably don't want to use this one. And on to the last one. Also decrease the CV, so this is something we want to use. Now that we have our internal standards, which are all of them except the PE, we choose same lipid structure and we normalize. So you can see there was a decreased CV in the QCs. It is also important to keep an eye on the sample so that their CV doesn't increase drastically. Now we save the results and we remove the internal standards. If we go back to our sequence panel, we can see that we included information on the sample amount, so we will perform sample amount normalization. You can see that these values are not that different from each other, so this will not affect our data too much, but when there are bigger differences, this is a very important step in the workflow. Now we will do drift correction. As stated in our documentation, this requires the information in the column order to be complete for the samples and the QCs. But before going through with this, we need to make sure that the QCs don't have missing values. And if they do, we need to impute these values. And we can see here in the summary that the QCs have missing values, so we will impute by making sure that only impute QCs is selected. And now we don't have missing values. There are two algorithms available for drift correction. We will go with the random forest and with the standard number of trees of 500. But for more details on this, please go through our user manual. Let's go back to the PCA where we have our unsafe data on the left, and we can run PCA, and again we can see the lower CV in the QCs. If the variation increases a lot in the groups, the QC should probably be checked. There could be something wrong in the samples, and you should skip this step if that's the case. But this looks okay, so let's save our data. Now we repeat the same procedure for the negative ion mode, but we only need to upload the file and then we can click reuse sequence. We repeat the process for the negative ion mode. We will go through all the steps as before, including the internal standard normalization, where we went through them one by one. For the negative ion mode, we used only the first two internal standards. Once we are done with the workflow, we can go to merge and select our positive ion mode, and then we set the MC tolerance and the retention time tolerance, which are analytical setting dependent. Here we will set the retention time to 0.03 and we'll edit priorities to MS2, which means we will prioritize the features which were annotated. Here we look for MS2 in the name column. Here we have the results from the merging. And you can see that in the second cluster, the app prioritizes the MS2 annotation instead of the lowest CV feature. And then we can remove the duplicates and save this as a new file. After this, you can explore the data again just to make sure everything is fine. And we can run some statistics on it. We can do it either in the app, where we have multiple options. For example, this pair doesn't work for this dataset since we don't have paired samples. We can also select groups to export for polyS test, or as we're doing now, export the whole dataset to polyS test. And this app will detect our different groups. It takes a little bit to load, but Here you can see our 9 different conditions. On the left you can select the comparisons you want to make. Right now we are going to do a simple quick test for comparing condition 1 and condition 2. In the right corner of the screen you can see the progress of the test. It can take some time, this frame is sped up, but once it's done you can see the results and you can export them to a CSV file. And this is an example of how we can integrate polyS test into our workflow.